However, before students can start generating their own data and perform experiments, they have to learn proper cell culture techniques. And this is also something where the Lux2 can contribute. One very essential thing when we want to perform experiments is that we have a healthy cell culture, and a healthy cell culture means that you also have to passage your cells at the right time or subculture at the right time. So if we look, for example, at the growth curve of cells, we identify different phases. First of all, we see the lag phase, followed up by the log phase, then the stationary phase, and the dead phase. What happens in the lag phase is that cells are still adapting to their culture environments. So, for example, the amount of growth factor that is present, but also the temperature or the pH that they are surrounded by. And after the cells are adapted to the culture conditions, they will enter the log phase. And this is the phase where cells actually start dividing and start growing. In this phase, you will also see an exponentially growth in the confluency of your surface. And in this phase, it is also assumed that the cell has the highest viability. So when you want to perform experiments or passage your cells, this is the right time to do it. And because overcrowding of passaging your cells too late can lead to overcrowding, apoptosis, and also senescence of your cells. And that is when they enter the stationary phase. This phase is characterized by a percentage of 0 to 10% of cells that are in their active cycle. So when you want to perform experiments with your cells, this is actually too late. And it also makes sense that this phase is followed up by the dead phase and your cells are useless. So what students can do with the Lux2 is that they can culture their cells, and via the automated image analysis they can obtain such a growth curve of their cells. And in this way they can really identify different phases along the growth curve. So they also know when they can perform experiments with their cells, but also know when to passage them. And in addition to that, they can also look into the effect of differences in culture environment. So as I already mentioned, for example, the different growth factors that are surrounded, the pH or the temperature of the culture. Because different cells also need different culture environments. So for example, some cells grow better at 25 degrees than at 37 degrees. And in this way, students can see what is the best for their cell type. So for example, they can seed cells at 25 degrees, but also at 37 degrees, and look into the difference in the growth pattern of both cultures. Now what you can see here is that these cells grow much better on 37 degrees than at 25 degrees. So when you want to perform experiments, this is really a crucial thing to investigate. Another thing that students can look into is the cell density, because cell density really has an influence on, for example, gene expression levels, and especially in a field like tissue engineering, for example, these expression levels are used to identify certain molecules that are involved in pathological processes. So what students can do, they can grow cells at different densities and eventually again look at the difference in the way that these cells grow. And what you can see here, for example, is that the culture that has a higher seeding density grows much faster than the cells in the lower density. And this is also identified in the growth curve that you can obtain out of these time-lapse videos, where you can see that in the higher seeding density, after 72 hours, the cells have reached a coverage of around 90%, while in the lower amount of cells, the cells have only reached a coverage of 50%. So this definitely makes some difference when you want to perform experiments. And the last thing, but definitely not less important, is that contamination can be identified and discovered by students. So they can really start an experiment, induce contamination, and they can learn how to identify, for example, a fungus growing in your cells. And this also saves a lot of time when they get to perform experiments in the future, because now they can see from home after 6 hours of culturing that contamination has taken place, instead of going back to the lab after 3 days and then seeing that contamination has taken place. So these are just some examples how the Lux2 can really improve the cell culture techniques, and in this way students will also have more confidence when they are working in the lab. And this also leads to more motivated students to eventually also get a better engagement of students. But next to that, the Lux2 can also contribute to the way in which students understand dynamic processes. For example, a process like cell proliferation, which is currently taught in lectures by showing images, where teachers explain how cells divide, how cells grow, but also how cells proliferate, which is basically a combination between cell division and cell growth. So with the Lux2, students can really see their own cells and identify different processes in their cell culture. So they can really see different cells dividing, but also cells growing, and eventually follow the proliferation pattern in their cell culture. And what is also very important about this video is that they can really access it from anywhere. So for example, show it to their parents or, as I said, their sister or brother. And it makes such a video much more valuable than any random video on YouTube, for example. Another concept that students can look into is cell aging, because cells are known to age and eventually become senescent, where the cell loses its ability to divide. And that is because the telomeres shorten. And these telomeres are short patterns of genetic material that protect the end of chromosomes. So it really protects the rest of your genetic material. So when these telomeres are shortened, you can imagine that the way these cells respond to certain external stimuli is really different when you compare them to cells that are younger. So in this way, students can make two cell cultures, one with young cells and one with senescent cells, and investigate the difference of the cellular response upon a certain stimuli. And what is also a very important mechanism to look into is, for example, cell death. In lectures, it is taught that cells can die in two different ways. First of all, apoptosis, which is a regulated form of cell death and is really necessary for an organism to stay alive. And they can also die via necrosis, which is an unregulated form of cell death. And it only happens when cells are exposed to extreme culture conditions. And eventually they are also taught how they can identify apoptosis and necrosis, for example by detachment of cells, by forming of blebs, but also vacuole formation or multinucleation. And what you can see with the Lux2, which I'm going to show in the video on the right of the screen within the red circle, is that you first see a cell detaching from the surface and eventually these blebs are formed. So when you see this process actually happening in a video, it makes much more sense than when I tell you this by showing these pictures. And that also applies to a process, for example, like cell migration. Because students are taught that cells can migrate upon different mechanisms, for example, by a difference in chemoattractant gradient, a difference in rigidity, or a difference in density of adhesion sites. And again, this process makes much more sense when you actually see the cells moving upon a certain stimuli instead of just looking at the pictures on the right.